With the right genetic treatment, even the simplest organisms can be engineered into massive monsters. Here's how to synthesize these 6 distinct amphibious anomalies. Initial specimen, the Dualottle. Why select the Axolotl genome? Utilizing the Axolotl genome as a basal template provides an ideal foundation for the Dualottle, given their profound phenotypic similarities. First, we must isolate and reduce its entire somatic framework down to its core genomic sequence. Genetic modification is strictly limited to macro scaling. The elongation of the anterior limbs, and inducing a bicephalic predisposition for the secondary head. Consequently, internal organ systems must adapt to regulate the heightened metabolic demands of this expanded somatic mass. Genome sequencing has reached successful completion. The CRISPR-engineered organism, designated the Dualottle, an erect, bicephalic, macro-scaled axolotl variant, has finally been fully synthesized. Genetic synthesis difficulty rating, 30%. The organism designated Hear Me Out monster is highly anomalous, requiring a chimeric hybridization of Anuran and Gastropod genomes stripped to their core profiles. We excised the Gastropod's shell synthesis genes prior to genomic integration, modifying the sequence to induce macro-scale gigantism truncate the elongated Anuran hind limbs, and activate caudal development. This specimen features 2 distinct, fully fused internal organ and neural systems. Sequencing for the Hear Me Out monster is finalized. Genetic synthesis difficulty rating, 50%. Next, the crab Titanic. This macro crustacean, utilizing an integrated vessel as exogenous armor, requires a basal Brachyuran genome. We must reduce this genetic profile to its fundamental core. Modifications are introduced to induce immense macro scaling and adapt the carapace for external compatibility. While analogous to hermit crab behavior, this monumental scale requires anchoring a full-sized metallic ship. Sequencing for the colossal crab Titanic is complete. Genetic synthesis difficulty rating, 15%. Next, the Starfish sea monster, a colossal echinoderm utilizing a standard asteroidea baseline, stripped down to its fundamental DNA. We introduce modifications to induce extreme gigantism. And restructure its dietary apparatus into a highly centralized oral cavity at the ventral core. To support the expanded somatic mass, fast-twitch muscle tissue is integrated. Engineering of the Starfish sea monster is complete. Genetic synthesis difficulty rating, 10%. Next, the specimen designated, "We swam too far." This humanoid sauropod utilizes a Brachiosaurus genomic baseline. Macro-scaling modifications are omitted. Instead, we suppress cervical development to yield a truncated neck alongside a vestigial caudal structure. The primary complexity involves extensive cranial restructuring to achieve a hominid morphology. Requiring the splicing of human DNA to express anthropomorphic organ systems and cutaneous tissues, successfully overriding the dense sauropod hide. Engineering is successfully finalized. Genetic synthesis difficulty rating, 30%. Next, our most legendary specimen, the sea eater. This colossal, two-limbed leviathan requires an amphibian genomic baseline to leverage their basal four-limb architecture and aquatic viability. After isolating its genetic core, we excise the hind limb development genes and heavily amplify anterior limb elongation. We then replace the standard amphibian skull with an agnathan jaw structure derived from lamprey DNA. Lastly, we trigger macro-scale gigantism and upscale the visceral organ systems. Engineering successful. Difficulty rating, 45%. From the bicephalic Dualottle to the legendary sea eater, these 6 specimens prove that with the right genetic protocols, no biological blueprint is set in stone. The sequencing is complete, the DNA is locked, and the era of engineered megafauna has officially begun.